Thank you for this kind introduction. And I rarely have a good excuse for giving a bad lecture, but this morning I woke up with high fever. I have the flu, and it's interesting over these 20-some years that we've been working at deCODE, we have been trying to work on the genetics of vulnerability to infections, and we have very little to show for it except a few common variants that associate with risk of tuberculosis. One of the studies we aspired to do was to look at the Spanish flu from 1918, because the history of the Spanish flu in Iceland has been documented so clearly. There were 487 people who died from the Spanish flu. We know exactly who brought the flu to the country. There was a singular person who left a ship who came to the harbor. And we could impute sequence variants into a very large percentage of these people who died. And the idea was to figure out why did some people die from it and others didn't. Because people who died from the flu, they died from hemorrhagic fever. So the influenza virus has a neuraminidase, and when the neuraminidase cleaves off the terminal sialic acid from glycoproteins on the surface of platelets, they are taken up by the asialoglycoprotein receptor on hepatocytes. And we looked and we found absolutely nothing. What I think is, however, interesting is that we found a variant in the asialoglycoprotein receptor that protects against heart attack. So Amgen, a pharmaceutical company in California, developed inhibitory antibodies against the asialoglycoprotein receptor, and now I have this passion to try to convince them to use it in the treatment of hemorrhagic fever in diseases like Ebola, etc., which is also, where patients also die from hemorrhagic fever because of thrombocytopenia. But that doesn't change the fact that here I am standing before you, an old, sick man, and I hope I will be able to stand here for the next hour. I'm not entirely sure. And you make sure that I will be carried out in a body bag if that happens. So what we have been doing in Iceland for the past 20-some years is to try to figure out how variation in the sequence or diversity in the sequence of DNA causes human diversity. We have been working like everyone else. It is relatively simple. You work on a data set on diversity in the sequence and data set on diversity in phenotypes, and you look for non-chance association between data points in the two data sets. And if your data set is large enough, it's amazing what you can do. It's amazing what you can overcome when it comes to incompletion in your data. And we have been mining this diligently and have come up with an awful lot of associations. And these associations basically fall into two major categories, the common ones that put us on a normal distribution curve of physiological function, and the common variants in the genome are responsible for most of normal human diversity. And then you have the rare variants that disrupts the same physiological function. And we, like everyone else, we worked on the common variants when we are before the sequencing capacity, or the capacity to sequence large number of people became available. And of late, we have sequenced now the whole genomes of 50 to 60,000 Icelanders. And once you begin to look at the rare variants, your experimental approach changes. And a good example of that is in work that we did on a family in Iceland, where schizophrenia and diseases related to schizophrenia were common. We found a loss-of-function mutation in a gene that makes an RNA-binding protein called RBM12. And this was a loss-of-function mutation that got rid of one of the three RNA-binding domains. And we had this observation for several years and couldn't take it any further. We couldn't find any other variants in this gene that associated with anything until we found in a public database a patient with a loss-of-function mutation in RBM12 from Finland. And we chased down the scientist who had come up with this variant in Finland, and he turned out to have a family with several individuals with another loss-of-function mutation in RBM12. So here we had it. And I think that this is an example of what we will probably have to face, we will always face in the near future, where we are working on variants that are very private to individuals, to families, and particularly to populations. So one of the things we are doing now, or one of the things that has changed in our experimental approach, is that when we are looking at very rare variants, we have abandoned the case control experimental design, and we are focusing very much on families. We are focusing on not just looking at association between diversity in the sequence and diversity in phenotypes, we are also looking at the changes in diversity. And one of the things that has spurred us on in doing that is that we have we sequenced the very last number of Icelanders. We have a fairly good idea of the sequence diversity in the Icelandic population, and we have started to do clinical sequencing for the National Hospital of Iceland. And the only thing I wanted to point you to on this slide is the fact that 1 out of 10 children born is born with a de novo loss-of-function mutation in a gene. And that means that 1 out of 20 children born is born with a loss-of-function mutation in a gene that is only expressed in the brain. And we have, we have done all kinds of things with this data collection. We have, for example, we have about 20,000 individuals who are homozygous for a loss-of-function mutation in Iceland, and we have a phenotypic center where we are recruiting these people, etc. But the thing that has has probably had the biggest impact on what we are doing is our work on the clinical sequencing. So we are doing clinical sequencing for the National Hospital of Iceland, and we are not selling this. This is done as a service to the population that has empowered us to do all of this genetics. And I'm just going to give you one example of a case that we have gone through just to show you how we are going about this. And this was a relatively young male with cervical dystonia and feeding difficulties, contractures in hands and feet, and also involuntary movements of extremities and subtle intellectual disability. So we whole genome sequenced this individual and his parents, and basically found about 5 million variants. There are 17,000 coding and splice. We used a frequency filter that is based on somewhat arbitrary. For example, we didn't want, when we are looking at the homozygous variants, we don't want to have anything that is more common than 2%, because that's dictated by the most common recessive disease in Iceland, which is cystic fibrosis. We found 3 variants that met this filter and were absent from the parents, and one of them has been known to cause dystonia. And so what is happening when we are doing the sequencing is that we have to continuously scour the literature for variants that are being reported to associate with particular disease. So this is pure clinical sequencing. We are not in this work basing our conclusion on our own discoveries, but we are basically trying to figure out into what group the individual fits that we're sequencing. And there was a 3-base in frame deletion. It is an area of the protein that is very important for its function. And basically, when we look at the clinical picture is very similar to what has been described before. And it is astonishing when you look at the cases that we have sequenced of children with peculiar syndromes. It's astonishing how large percentage is accounted for by de novo mutations. And then the big question is, what happens to these de novo mutations? How many of these de novo mutations that have functional importance will be carried on to another generation? And I will come back to it in a little bit later. But we have been, and this clinical sequencing has at least had a significant impact on me and has turned my focus again to things like recombination, gene conversions, and de novo mutations that we have been studying in the past. And I cannot resist to show you sort of our first foray into the study of recombination. And it is, I'm just taking an advantage of the opportunity to thumb my chest. And we basically put together what we called a high-resolution recombination map. It was based on only 1,257 meioses, all right? And it basically showed us what we all of us know today, that the recombination peaks and valleys do not always coincide when you look at men and women, which is very important to keep in mind because it is clear that the location of recombination hotspots is dictated by more than PRDM9. It is more than just the sequence that dictates it because of the difference between recombination hotspots in men and women. But what was interesting in this is that we developed this map when the sequencing of the human genome had not been completed. And it's interesting because you had all of these great minds of human genetics who went on to try to sequence the human genome, and they forgot to put together a genetic map because you can really not accurately assemble a genome without having some sort of a genetic map. You have to figure out which pieces travel together. And it's interesting, this is just a demonstration of the fact that there was a very large piece that was incorrectly aligned before we published our map. And what's particularly interesting is that when it comes to placements of SNPs in the public sequence, that before we published our paper, it was only 92% accurate. After we published our paper, it became 99% accurate. And the guys in the trenches working on the sequencing sent us a large number of emails thanking us for the map. So we made a minor contribution to the sequencing stuff. Soon after that, we also published a paper on recombination rate and reproductive success in humans. And here I have to apologize because there is absolutely no correlation between reproductive success and recombinations. And the way in which this works is that recombinations in women increase with age. The older the mother is when the child is born, the larger number of recombinations she hands over to her offspring or child. And we saw that the higher recombination rate, the more children did the woman have, but that is simply because she had children into older age. So she had more children if she had children when she was older. And this wasn't obvious when we had the data that we had then. When we had the data that we had at that time, there was a significant correlation, even if you corrected for that. But subsequent data have shown that our conclusion was in error. But it is important that the recombination rate increases with the age of the mother. It isn't entirely clear why. The double-strand break happens when the woman is at the fetal stage, but the resolution of the double-strand break happens just before ovulation, and we don't know, in a way, we don't know which mechanism it is that is affected by age. That has yet to be figured out. We found variants in the genome, in the RNF12 gene, that are fairly interesting haplotypes that increase recombination rates in women, decrease in men, and vice versa. So it's not just that there is a different location of recombination hotspots in men and women. There is a difference in the control of the rate of the recombination in the two sexes. And it's interesting also if you look at it that histone modification have a very important impact on recombination, and there are very few recombination in the transcribed part of the genome. But there's an interesting recombination pattern differences between men and women around genes. There is a relatively high rate of recombinations within large introns in men, but not in women. But flanking the genes, about 50 kb away from the 5' and 3' end, there is a high recombination rate in women. So basically, the recombination coming from the men shuffle the exons, the recombinations of women shuffled genes, all right? And I will come back to the recombination frequencies around genes when I discuss a new study that we have recently done. We have also studied gene conversion, the non-crossover recombinations or gene conversions. And it's interesting because it's interesting because the pattern that you see there is not all that different from what you see with recombinations. When we are studying the gene conversions, we have set certain limitations. For example, we demand that we are working with haplotypes that are at least 2.5 centimorgans, and we stay about 400 kbs from the telomeres. But what you see there is that you see also an increase in the maternal gene conversion rate with age. And actually, the increase in the recombinations in women, or the maternal recombination, is very subtle. So if you take a mother, a 20-year-old mother versus 40-year-old mother, there is only a 4% increase in recombination. That amounts to about 2 recombinations, additional recombinations, after these 20 years of age. However, the age-related increase in gene conversion is much larger. There is about 2.5 times larger number of gene conversion in the 40-year-old versus the 20-year-old mother. The gene conversions frequency is highest by telomeres, which means that the gene conversion rate is greater in the short chromosomes than the long chromosomes, etc. But the change in diversity that matters the most, the one that has the biggest clinical relevance, is of course the de novo mutations. And at the end of this presentation, I'm going to show you that the recombinations and the de novo mutations are not totally independent of each other. But we have studied de novo mutations for a few years, and it is interesting, when we started off looking at de novo mutations, the big question in our mind was, what is it that accounts for diversity in the rate of de novo mutations? Is it environmental, or is it genetic? Or if it's genetic, what is it? And basically, about 97% of diversity in de novo mutation rate is related to the age of the parents. 80% of it from the father, 20% of it from the mother. That's interesting because the concern then on population level for the environmental contribution is relatively little. When you sort of walk along the chromosomes and you look for peaks and valleys in de novo mutation rate, there is really nothing remarkable to see in the paternal de novo mutations. But when you look at the maternal mutation, you see, for example, towards the short arm of chromosome 8 in the mother, there is a dramatic increase, or dramatic peak in recombination rate. So you have these peaks in the maternal genome where the mutation rate is fairly high. It is not in the paternal genome. And actually, most of these mutations happen to be GC mutations in these peaks, all right? And what is interesting, if you then go and you look at sequence data like in the 1000 genome, this is in the regions where there is high density of GC SNPs. And these are high density of GC SNPs with various frequency of the minor allele, which indicates that these regions are regions where there seems to have been a peak in de novo mutations for a long time. It's interesting that you will see that there is that you will see a string of complex mutations more frequently coming from the mother than the father. And what you see actually in these regions here is that you will see that there's a strand concordance in this mutation in these regions, which is about 10% of the genome. And it's interesting that in the maternal genome, this happens to be a region where there is an age-related increase in de novo mutations. And elsewhere in the genome, the age-related increase in de novo mutation is much, much higher in the paternal genome than the maternal genome. But in this place, they are almost equal, all right? And you will find almost equal number of de novo mutation in this region coming from the mother as comes from the father. So you have almost twice the number of de novo mutation there than you have elsewhere in the genome. And what should that mean? It probably means that whatever attributes are conferred on us by genes in this region, they should probably evolve a little bit faster. One of the, so this is about, these regions constitute about 10% of the genome. And this telomeric piece on the short arm of chromosome 8 is a very good example of that. But we went ahead since it looked like that is whatever vulnerability on this part of the genome seemed to be old, we went to the phylogenetic tree and we basically demonstrated that in the chimpanzee and the gorillas, these regions have approximately the same quality as in man, but in orangutans they don't. So this region seems to have become vulnerable, probably to double-strand breaks, since the orangutans and the others separated. We have looked diligently to search for attributes conferred on us by genes in this region, and of course we began by looking at genes that are expressed in the brain, but we have completely failed at figuring this out. But it's particularly interesting because it's not just that this is a region where de novo mutations, age-related de novo mutations, occur in the maternal genome. The increase in recombination rate also happens mainly in this region, and also the increase in gene conversions. So this 10% of the genome constitute a region that is particularly vulnerable. The de novo mutations, of course, are, as I said before, very important clinically, and had led to all kinds of jokes about their old father, etc., because of the increased load of de novo mutation coming from the old father. And the fact of the matter is that a 40-year-old father is a child that is conceived by a 40-year-old father, a 40-year-old man, is 2 to 3 times more likely to develop autism and schizophrenia, and then we have all kinds of course, rare cancers of childhood that have higher incidence in children conceived by old fathers. And we have actually fairly interesting sort of demographic data from Iceland where we can demonstrate how the mean age of fathers at the conception of their children has changed over the years. And there was a dramatic decline in it in the beginning of last century, and then steep increase again around 1970. And there are those who have speculated on the connection between that increase and increase in the diagnosis of autism. I think that is stretching it a little bit. We have started, and we are just writing up, a very large study on recombinations. And we have finished the analysis. We are writing up a couple of papers on it. And as you can see, this is based on 126,000 meioses. It's about 100 times larger than the study we published in 2002. It's not just the number of meioses. We have now sequence data, sequence level data on 21,000 meioses. And we are using the genotype data to look for phase changes. We are refining the crossovers by using the sequence data set and use EM algorithm to estimate recombination rates between each pair of adjacent markers. And the minimum distance between a adjacent crossover is 2.5 centimorgans. And this is just showing the normal diversity in the number of crossovers. And the resolution of this map is less than kilobase. It's about 680 base pairs, which is an incredible increase in resolution from previous maps. And our map is about 400 kb from the telomere, and the overall length in the map is similar to what has been previously estimated. The differences are subtle and probably not of great relevance. Due to the high resolution of our map, our recombination hotspots are also smaller, all right? And the size of them currently is approximately the size of the double-strand break regions. And we can, because of the resolution, we can show some recombination hotspots that are clustered together. So previously they looked like one hotspot, but here actually a clustering of hotspots. And why is the resolution important? It gives us better estimate where crossover occur. It allows us to identify sequence attributes important for crossover. And it has also allowed us to find mutations close to the crossovers. And if you look at chromatin modification, there is clearly an underrepresentation of recombination in transcribed regions and overrepresentation in enhancers and promoters. And remember, the crossover occur early in replication timing, and they occur where there is a high GC content, and the paternal crossover occur close to the telomeres. And as I said before, the recombination rate increases with maternal age, gene conversion rate increases with maternal age, and mutation rate increases with maternal age, and it does not happen uniformly across the genome. It is mostly focused on this 10% of the genome where you have the high de novo mutation rate. And as I said before, the increase in recombination rate that goes with the age of the mother is relatively subtle. and remember, there is an increase, there is about 3 times greater increase in recombination rate in this 10% of the genome versus the rest of the genome, just in this, the 10% that I showed you before. And there is, with increasing age, there is a shift towards later replication regions, and the GC fraction diminishes with the increasing age of the parent. There is people have been talking about correlations between mutations and crossover, and it's been well known, but it has not been directly shown so far, all right? And it's not been clear whether the site of recombinations or mutations share sequence attributes or whether the recombinations themselves are mutagenic. But we have been able to study this in detail, and we can show that sort of on 1 kilobase on both sides of the recombination spot, the mutation rate in males increases by 39.5 times, in women 39.5 times, in men about 56 times, so it's on average about 50x increase in mutation rate. And what's also interesting that if you extend this to 100 kb on both sides of the recombination spot, there is an increase in mutations in the maternal genome, not in the paternal genome, and that is mostly where you have complex crossovers, where you have recombination plus gene conversion in the same spot. So this is of importance. If you're only looking at this very, very high increase in recombination rate, that should not be particularly meaningful when it comes to the transcribed part of the genome, because the distance is too large, except that you will have a mutation in enhancers and promoters, and that may have significant impact on gene expression. There is a difference in the cancer mutation, which nucleotides are switched depending on sex and whether or not there is it's close to recombination site. In addition to this, we also looked at, still another time, for variants in the genome that impact rate or recombinations, and I'm not going to go into the details of that today. We're sort of polishing it. But we have found a very large number of new loci with very large effect, and some of them are in genes that you would expect would be relevant when it comes to recombinations. We have done more than that. We looked at sequence features that are associated with recombinations like DC fraction, replication timing, and distance from the telomere. We created phenotypes from that, and we looked at association with those phenotypes. And remember, this is an attempt to understand in more detail than PRDM9 gives you. So how is the area selected for recombinations? And we have found a very significant number of associations, and some of them with fairly large effect. And I'm not going to go into the details of that now. But one of the things when you're doing clinical sequencing, and in particular when you're doing clinical sequencing in a small community, you always end up by getting phone call from anguished parents who have a child with a devastating condition because of a de novo mutation, and they ask you, what is the probability that this would happen to a subsequent child? Would our children, subsequent children, be at greater risk of developing a disease? And we launched a study into this specifically after one of these mothers had phoned me, and we basically took about 1,000 sibling pairs from 253 families. And these were some of them very large families. This one family, there were 17 children. There were 500 out of this 1,000 sibling pairs, 500 of them came from families with more than 8 children. All right? And we found, and you realize that although 80% of the de novo mutations come from the father, the ones that are shared by siblings, they come approximately equally from the mother and the father. And there are all kinds of explanations to that, and one of them is that the spermatogonia continue to proliferate throughout life. The lineage of the ova is much shorter, etc. But one of the things that we have done is that we have taken the de novo mutation in these large families, and this is from a family with 10 children. And basically, when we looked at the de novo mutations, both coming from the father and coming from the mother, they fall into 2 clades. All right, and why should that? Why should they? And they always fall into 2 clades. I don't know why they do so, but you can postulate that in the case of that one of the clades come from the left testis and the other from the right, etc. There are all kinds of reasons for that. But one of the things that you have to recognize is that the de novo mutations that we say are not present in the parents, and therefore we call them de novo mutations, they are mutations that come from the parent. And what is important here is whether the mutation takes place before the primary germ cell specification or not. Remember, after the specification, that the germ cells migrate to the gonads where they proliferate, etc. If, however, and one of the problems when you begin to look for de novo mutations, and we were so subtly guilty of that crime, is to use a filter that says if we find any, if we find any reads in the parents, it's not classified as a de novo mutation, all right? But if that is, thereby you throw out an awful lot of the early mutations, because if the mutation happens before the primary germ cell specification, you will see somatic mosaicism in the parents, all right? So one of the things that we did looking for these mutations is that we did not just take the ones where we set the filter that it was not supposed to be in the parent, but we also took siblings who shared the haplotype but did not share the mutation. So you have to use all kinds of other means, and basically, if you take mutations, de novo mutations that are shared by sibling, you have about 52% probability of finding reads in the parent from the parent's genome, or show evidence of somatic mosaicism. And actually, one of the interesting things is that with increasing age of the parents, the percentage of the mutations that are going to be sibling shared decreases. They are simply more so in the father than the mother. The absolute number of the shared mutation is going to remain the same, but the de novo mutation number increases so dramatically in the male, so there is about 2.4% decrease of probability of the sharing with each year in the case of the male, 1.7 in the case of the mother. But basically, this is just an example of our case where there's a mutation in the syntaxin binding protein in young siblings with unexplained epilepsy, who both had the same mutation, and the allelic ratio in the father was about 17%. And what's interesting, if we looked at the grandparents, we don't find it there. So it's a de novo mutation that comes from the father. And when you begin to try to assess the probability of recurrence of a de novo mutation in a subsequent child, what matters the most is the parental somatic mosaicism, because it means that if you find it, it's probably happened before the primordial germ cell specification. What matters is also sharing by other siblings, parent of origin, mutation time, and genomic location. And the interesting thing is that you can put together a recurrence model that you can use to give a fairly accurate assessment in the higher risk category. And if you take the theoretical estimates that people are throwing around, it's that the probability is somewhere between 0.048 to 9.4%, but based on our data, it ranges from 0.011% up to 28%, which is dramatic difference. And when you are up to about 30% probability of the recurrence of the mutation, it becomes clinically very, very relevant. Then the question is, what happens to these de novo mutations? Most of them, like in the case I'm showing you here, they just come and they go because they prevent reproduction. The individual with the mutation is devastated. We were asked to sequence a young male with severe intellectual disability and recurrent seizure and dysmorphic features. And we found an insertion or a complex mutation in a gene that was absent from the parents, absent from 40,000 Icelanders. There was a frame-shift premature termination, and this gene is the heterogeneous nuclear ribonucleoprotein U, or scaffold attachment factor, or A. And what's interesting is that it is a gene with normal, almost normal, nothing remarkable when it comes to constraint. You will find approximately as many synonymous mutations as expected, approximately as many missense mutations as expected, but it's completely intolerant for loss of function mutation. So all of the loss of function mutations that have been found in this gene have been de novo mutation. An example of a mutation that only shows up in this context of the mutability is not the inherited mutation. Another diametrically opposite example is that we found the other day, and I'm just going to skip over the discussion of the LDL receptor, we we found a deletion, 2.7 kilobase deletion out of the 3' UTR of the LDL receptor, all right? And it was not a de novo mutation as where we found it, because we found it in a family. And interesting because it associates with dramatically low LDL. You know, LDL of 3 standard deviation lowering in LDL. But it is pretty clear that this mutation happened in a grandfather in this family. But, you know, there is absolutely no probability that this mutation will become more common, because we have destroyed that with statins and anti-PCSK9 antibodies. We are beginning to mess with evolution with our drugs. But there's more than just recombinations, gene conversions, and de novo mutation. Drift is also a force that shapes our genome, and it's probably the force that shapes it the most, at least in a small country like Iceland. And just want to remind you that Iceland was settled by Norwegians who stopped by in British Isles, picked up women, and went up to Iceland and settled down. And there's the Book of Settlement that was written 1,000 years ago that says that this is the way in which it happened. So we went and we looked at Icelandic Y chromosomes as markers of paternal lineage, mitochondria as markers of paternal lineage, and we showed that in modern day Icelanders, about 75% of the Y chromosomes are Norwegian, about 65% of the mitochondria are Celtic. So Iceland was settled by Norwegian boys, and we have actually new data showing that they did not come from here like the history says. They came from this part of Norway. They went to Norway, they picked up women and went up to Iceland, settled down. I'm not entirely sure that they all came with mutual consent. I think it is possible that the Me Too revolution would not have liked what happened there. But what is interesting is that we have subsequently gone and we have looked at DNA from skeletons from the time of the settlement of Iceland. And what is interesting when we looked at the current day Icelanders versus the Icelanders from the time of the settlement, and we compared the DNA from the time of the settlement to DNA from Scandinavia or Norway, Iceland, and contemporary British. And the only current day population that deviates significantly from one of the settlers is the Icelandic one. So we are least like the settlers. So we are less like ourselves than anyone else. That's strange, isn't it? Now, what this basically tells us that we have been changed more by living in this harsh condition for 1,100 years than you have been changed by all of this stream of people constantly coming through Great Britain. And a lot of that is rooted in, the fact that we went through dramatic population bottlenecks, etc. But then the question is any of that change clinically significant? And I think so. Let me give you an example. We did a large study of malignant melanomas. And when we started looking at malignant melanomas, there was one variant that had been associated with it, and it was in the melanocortin type 1 receptor, all right? But at that time, when we looked at it, the odds ratio in Iceland was 1. It had no impact on the risk of malignant melanoma in Iceland. The odds ratio in Spain was 2.56. In Sweden, it was 2.26. And remember that this mutation in the melanocortin type 2 receptor is the mutation that makes your hair red and your skin sensitive to sunlight, all right? And it's interesting that, if you live in Iceland, it isn't a particular handicap to be sensitive to sunlight. We do not carry sunlight in big buckets in Iceland. It comes in small containers. But what's interesting is that the frequency, allelic frequency, is 26% in Iceland, it is 6% in Spain, and it is 17% in Sweden. So I'm pretty convinced that the difference is not because of selection, it's because of drift. But the drift is not inconsequential. We have documented some unfortunate selection in Iceland that has also caused some changes in our genome. And it is the selection of variants in the genome associated with educational attainment. It shouldn't come to you as a surprise that it has been known for a long time that people with high education have usually fewer children than people with little education. And that basically means, in your language, that there is a selection against education. We took polygenic score for educational attainment, and we showed that people with high polygenic score for educational attainment have fewer children. And then even if you took just those with high polygenic score for educational attainment who do not have a lot of education, they just have the propensity, but they do not have the education, they also have fewer children. And then we showed that educational attainment is a very good surrogate for verbal IQ. So we are becoming dumber by the generation. And that means, I'm not sure you will agree with me, but that means that it is a privilege to be the oldest man in this room. But basically, what is sort of for me, standing here and talking to you in a conference on rare diseases, what has been sort of an eye-opener for me over the past few years is to look at the results of the clinical sequencing. It is so fascinating because just a few years back, I would never have thought that de novo mutations would be that important from a public health perspective, but it is. So thank you for listening. And I'm just proud that I'm still standing.
Thank you, Kári. We're very grateful that you're still standing and will not keep you here for much longer. But I'm sure your condition did not significantly impact your story, because it was a fantastic story. There's many different types of stories. Some are fiction and some are fact. And I think this was a scientific story, and we really enjoyed it. One question...
I think there was a subtle criticism in this. He was implying that some of what I said was fiction.
A good story always has some fiction, I think.
Where I come from, there's only one way to settle this.
Have a drink together. All right, thank you very much.
Any questions?
I have one question.
Yeah.
What was the book?
This is from the manuscript of a book of settlement.
So this describes how Iceland was
settled, yes. Yeah. And you have to realize that all of these old Icelandic sagas that were written somewhere between the year 1000 and probably 1300, they are probably all of them sort of historical novels in a way, but they fairly well reflect sort of the way in which these people thought about themselves, and all of them begin with page after page of genealogy. And that's one of our sources of the early genealogy. And that genealogy is actually fairly accurate. We have been able to confirm that that even though genealogy is fairly accurate.
There's a question here.
Richard Gibbs, Baylor College of Medicine. Of the 5 items, if you take away the SIB sharing and the parental mosaicism, do the other features still offer you some power to predict a second mutation?
Yeah, if you just have 2 SIBs who share, all right, then that gives you a very large percentage of the total possibility of prediction. It goes from 0.048 up to 23%. So you know, if you have parental mosaicism or 2 siblings who shared before, then you have gotten very large percentage of the total predictive possibility. That's absolutely correct.
Thanks.
I have a related question. So with your prediction of recurrence risk, that's all based on shared mutations that are not pathogenic. So I wonder whether you think about, when thinking about recurrence risk for pathogenic mutations, there's an additional factor, which is the fitness cost to the parent associated with having a mutation at a given level of somatic mosaicism. So for some disorders, 50% of cells may well be sufficient to give you the disorder, and therefore much less likely to have children. So one will therefore deplete high-level somatic mosaicism from pathogenic mutations in a way one may not from benign de novo mutations.
I think that was such a complex question. We'd ask you
No,
to summarize that.
It wasn't a question, it was a statement. Just, yeah, and I don't disagree with any of that. All right, this is just all of the data we have. And there is no question that if there is a somatic mosaicism, as we have seen, I gave a very large number of cases where we have found it. It gives us a fairly good accuracy in the prediction, all right? But it's probably going to become more accurate the more data we have. We are just starting off.
The story about the selection against IQ, basically, can you quantify for us what that actually means in terms of...
0.6 standard deviation per century.
Okay, and if you control for the actual level of education received, you mentioned that the effect is still there, but...
You have to realize that this is just genetic propensity. The thin effect there's more than compensate for that. And also you have to realize that we are probably dealing with relatively narrow range in IQ, and this may only be temporary. We only have, the data we have are data for about 80 years, and there isn't really a question that this goes up and down. But it's also when you begin to think about it, what is the role of how does the human brain, fit into history of evolution, all right? If selection has only to do with how many offspring you have, all right? And what does the brain that has given us the music of Bach, Mozart, and Beethoven have to do with the number of children you have? You end up probably have to look at the brain as an instrument that we use to cheat on evolution. We use the brain to help us to adjust to the environment without having to go through rounds of random change and selection. So the brain is a problem. It's not a problem in this sense, it's a problem because we haven't defined this idea how it works. In the year 2018, and you would ask me what is the function of the brain? It's to be an organ of consciousness, right? And consciousness has 2 components, major components. It's alertness that you lose and regain once a day. And then it is the content of consciousness, which are your thoughts and emotions. And our thoughts and emotions define us as a species, and they define us as individuals of the species. And I haven't the faintest idea how the brain generates a thought. Give me a definition of a thought, right? Philosophers have been trying to do this for centuries, and we have no definition of a thought. I'm completely
And you are aware that Francis Crick spent the last 10 years of his career trying to understand just that.
Yeah, poor fellow.
So maybe this is where geneticists gravitate over time.
Yeah, no, don't misunderstand me. I have great respect and admiration for Francis Crick, but not because of the results of his work on the brain. Any more questions? All right.
We've been inspired. We're very grateful. And when I gave you a prize many years ago at the European Society of Human Genetics, it was the Gilded P because of the Mendel lecture. Unfortunately, Jim Lupsky does not have a symbol, so I cannot give you something related to him, but I give you this as a small token, and thank you very much.